Dear students, our lecture today is about immune responses. In these three slides, there are some key definitions will be mentioned during the lecture. The learning objectives for this lecture are basic rules of immune system, dealing with bacteria and viruses in the extracellular space, dealing with intracellular pathogens. How does the immune system being alert to the presence of infectious agents inside the cells, and the terms antigenicity and immunogenicity. Basic rules of immune system. First of all, what is the immune system? Immune system, cells and molecules that contribute to immune defenses against infectious and non-infectious conditions. Self versus non-self. The first step is recognition step. Recognition of foreign substances and microorganisms that have overcome the outer defenses, just like skin epithelium, mucosal surfaces of the gut, respiratory and reproductive tract. The critical role here of the immune system is to determine what is foreign, non-self, and what is normally present in the body as self. Therefore, the cells and molecules that comprise the innate immune system are preoccupied with detecting the presence of bumps. What is bumps? Bumps is pathogen-associated molecular patterns, particular molecular patterns that are typically associated with infectious agents. The bumps, after being recognized, trigger activation of the innate immune system. The recognition of non-self or infectious agents, as in this figure, is done by certain molecules, soluble receptors, pattern recognition receptors, or cell-associated pattern recognition receptors on the immune cells. Pattern recognition molecule or receptors present either as cell-associated receptor PRR on the surface of immune cells like tool-like receptor, node-like receptor, and REGL1-like receptors, or soluble molecules just like complement, mannose binding lectin, C-reactive protein, and lysozyme. The pattern recognition molecules can recognize the bumps which are present on the pathogen. We mentioned complement. What is complement? Although we have specific lecture for the complement, but let's just define what is the complement. Complement comprises approximately 30 circulating and membrane express protein play a major role in defense against many infectious organisms as part of both the innate and antibody-mediated adaptive immune response. In recognition step, after pattern recognition molecules recognize the bumps of present on inside or outside the pathogens, then many immune steps or immune stimulation occur after that like binding the microorganism by soluble PRR molecule, enhancement of phagocytosis of PRR-bound BAMs, proteolytic cascade resulting in lysis of microorganism will be occur. Phagocytosis of BAMs and associated microorganism through the phagocytic cells, activation in immune cell countering BAMs, and release of cytokines to amplify response. The second step is elimination step. Elimination of these agents by a diverse repertoire of cells and molecules that act in concert neutralize the potential threat. Third step is memory. Creating a memory in the immune system in that case. Immune system is able to learn from encounters with pathogen and to maintain a reserve of cells of the cell that are able to respond swiftly to a new infection with previously encountered microbe. Immune system have acquired adaptive an ability called immunological memory. The immune regulation, the immune checkpoints. It's a number of immune regulatory mechanisms exist to ensure that immune responses are proportional to the level of threat that a particular infectious agent possesses, as well as to ensure that immune responses are not being directed against self and that responses directed only against non-self, and stop when the infectious agent has been successfully determined from the body. In this slide, the immune system will response of against the apoptotic cell and necrotic cell differs in that case. In apoptosis, apoptotic cell which undergo apoptosis program cell death, which display molecule on their plasma membrane that mark these cells for removal through phagocytosis before they can rupture and release their intracellular content. Apoptotic cells do not activate the immune system in that case, while the necrotic cell during severe injury where necrosis occurs to these cells, which caused by tissue trauma, burns, certain toxins that lead to rapid swelling and rupture of the plasma membrane of damaged cell. In that case, DAMs will be released. The DAMs, during the rupture of necrotic cell plasma membranes, danger-associated molecular patterns called DAMs will release and activate the immune system. Soluble pattern recognition receptors and cell-associated pattern recognition receptors will recognize these DAMs and bind with these DAMs in order to stimulate the immune system. Now, how the immune system deals with bacteria and viruses in the extracellular space, that's means outside the cells. For both pathogens, bacteria or virus, immune system recognize these agents by complement. Complement is approximately 30 circulating and membrane expressed protein play a major role in defense against many infectious organism as a part of both innate and antibody-mediated adaptive immune responses, and through acute phase proteins, antimicrobial peptide, they remove them by destroying them through membrane lysis. The same classes of protein involved in decorating infectious agent for recognition by phagocytosis by phagocytic cells, just like macrophage, neutrophil, molecules that are involved in the decoration of infectious agent to prepare them for removal called opsonins. That means prepare for eating. I think you already knew how phagocytic cells deal with extracellular pathogen in previous lecture and how to deal with bacteria and phagocytosis process occur. Here are some types of cells, just like macrophages, which present in all early organs, ingest and destroyed microbe in process of phagocytosis, present microbial antigen to T cells in adaptive immune response, and produce cytokines and chemokines. The monocytes present in the blood, 5 to 7% in the blood, recruited at infection site and differentiate into macrophage after that in order to kill the microorganism. Neutrophils present in the blood, 60% of the blood leukocytes increased during infection to kill microbial agent recruited by chemokines to the site of infection, ingest and destroyed biogenic bacteria just like Staph aureus, streptobiogenes. This figure shows types of opsonin and how opsonin act on the pumps present on the microbe. Here, the microbe displaying pumps on its surface will be recognized by protein pattern recognition receptors on phagocytic cell and bind with pumps. Also, there's many opsonin will bind with opsonin receptor on phagocytic cell. Opsonins include complement protein C3b, C4b. Also, antibodies act as opsonin, IgG, IgM. Acute phase protein like C-reactive protein, which is a protein made by liver, its level in blood increased during inflammation. C-reactive protein test measure the amount of C-reactive protein in blood to detect inflammation due to acute conditions or to monitor the severity of disease in chronic condition. C-reactive protein here act as opsonin for the microbe. Also, one kind of acute phase protein is the mannose binding lectin. The mannose binding lectin proteins found in circulation bounded with proteases known as MASP. They can bind with pumps, leading to activation of MASP in order to activate the complement system. In that case, it's just another pathway for elimination of pathogens. or when they bind with the pumps of the pathogen, the phagocytic cells through its opsonium receptor will bind with this connected receptor and phagocytes the microbe, in that case to eliminate pathogen. Dealing with intracellular pathogen. When the pathogen inside the cell, once the virus enters the cell, the proteins and phagocytic cell no longer be of any use in dealing with this type of infection because proteins cannot freely diffuse across the plasma membrane of the infected cell to either lyse or tag the infectious agent for phagocytosis. Although some specialized phagocytic cells, macrophage, can kill intracellular bacteria that have invaded them, most cells cannot do that very effectively. So how the immune system deals with viruses in that case? Some phagocytic cells have a role in killing intracellular pathogen through the oxygen-dependent pathway, respiratory burst, and oxygen-independent pathway through lysozyme, lactoferrins, transferrins, and other proteolytic and hydrolytic enzymes. How does the immune system being alert to the presence of infectious agent inside the cell then? There are a number of intracellular pattern recognition receptors can detect pathogens that have entered cells. This results in production of signals like cytokines and chemokines are proteins involved in signaling and communication between cells of the immune system. These proteins serve in chemoattraction, phagocyte activation, and in inflammation that alert the immune system to the presence of an infectious agent inside the cell. There's a clever way to displaying the breakdown product of pathogen after being phagocytosis to cells of the immune system through binding with MHC molecule involved in this process. MHC mean major histocompatibility complex that involved in displaying the breakdown product of pathogen on antigen presenting cell APC to the cells of adaptive immune system. It's also involved in transplantation. Displaying breakdown product by MHC to cells of immune system. This figure shows how dealing with either intracellular pathogen like virus in case of virus infected cell, or when bacterial cell engulfed by dendritic cell and phagocytosed and break down to small peptide. How these two mechanisms occur. In case of bacteria, when bacterial cell engulfed by dendritic cell or phagocytic cells and then break down to small peptide, these small peptides present on the surface of dendritic cell as a complex MHC class II peptide complex. Binding, that's mean binding the peptide with MHC class II on the surface of dendritic cell. This complex will be recognized by CD4 T cell. CD4 T cell, CD4 means the surface marker present on T cell. When binding with CD4 T cell, CD4 surface marker with MHC class II peptide complex, and then other receptors will bind together the CD28 on the CD4 T cell with its corresponding receptor on a dendritic cell or any antigen presenting cell B7, providing a signal. Second signal for activation of CD4 T cell in order to activate and proliferate, differentiate into helper T cell. In this pathway, the helper T cell will activate other immune cells like B cell, for example, to produce antibody, or activate other cells, immune cells, to eliminate the pathogenic bacteria. The other pathway, in case of intracellular virus, for example, in virus infection, dendritic cell, after in display peptide protein with MHC class I on its surface, and this MHC class I peptide, the small peptide with from the virus, break down the virus particles with MHC class I, it will form a complex on its surface, will be recognized by other type of T cell called CD8 T cell. CD8, also a surface marker on T cell, will bind and recognize the MHC class I peptide complex. And also it needs another signal, second signal, that means second two receptors from CD8 T cell and dendritic cell to bind together, providing a second signal to activate CD8 T cell. CD8 T cell, after activation, proliferation, differentiate into cytotoxic CD8 T cell. Cytotoxic T cell will, in that case, have the ability to kill the infected cell in that case. Antigenicity and immunogenicity. Antigen, any foreign molecule, non-self, can react with the product of the immune response. Immunogens, molecules that can induce an immune response. There's a very important note that not all antigens are immunogens. That means not all antigen can stimulate the immune response. Here are some immunological terms related to antigen. Epitopes. Epitopes, antigenic determinant, are small chemical groups on the antigen molecule that elect and react with the antibody. Hapten, it's a small molecule that is not immunogenic until it binds with larger carrier such as protein. In that case, it will elect the immune response. Adjuvant is a substance that enhances the immune response to an immunogen. Its chemically are related to the immunogen, causes slow release of immunogen leading to prolonged exposure to immunogen to immune cell. features of immunogenicity include fairness, molecular weight, chemical, structural complexity, dosage, route, and time of administration of antigen. All these affect the immunogenicity of an antigen. Factors contribute to immunogenicity just like genetic, age, immunosuppressed patient, lifestyle, type of diet, environmental factors. All these factors affect the immunogenicity. Thank you. Thank you very much.